and we wanted to welcome everyone to Renegade Research Roundtable. This is part 2 of the mechanical basis discussion. If this is your first time with us, you are very welcome. We're very happy to have you here. I am Isabel, the CEO of our organization. We are a nonprofit, patient and caregiver-led, decentralized 501c3, and we focus on infection-associated chronic illnesses such as MECFS and long COVID. Within this, we have 3 areas of focus: research that's agile and patient-centered, education to increase the number of clinicians treating us, and community, where we focus on practical advocacy around barriers to care. Most of you know us best from our largest project to date, Remission Biome. As a nonprofit, our work is funded by grants and private donations. Please visit the Remission Biome website if you are able to contribute. More importantly, even before we had any funding, our wonderful team of volunteers moved the wheels of our organization. All of us have a personal interest in advancing research and care of infection-associated chronic illnesses. To stay in touch with us and hear about upcoming events, please subscribe to our newsletter. And now I'll pass it out to our founder, Tess Fowler, to tell you more about today's roundtable.
Well, so I'll start presenting here. While I'm getting this up, my first question to everybody is, who was able to come to the first part of this roundtable in October? Raise your hands. Let's see how many we get. I can't see everybody right now, so I'll have to have somebody else get an idea here.
I'm looking at the participants or the attendees coming in, and their numbers are going up. So it's like 10, 11 are so far raising their hand, 12. So
okay, like somewhere between a third and a half.
Okay. All right, cool. So you can put your hands down now. So yeah, this is the second part of the conversation. These meetings came out of Ron Davis's meeting where he was talking about the molecular basis of MECFS. And we wanted to sort of try to connect different parts of the research. And one of the things that we think might be key is the Brainstem, and for some people, the mechanical basis model. So I'll just give a very quick recap of the first meeting, get people up to speed as much as I can before we jump into part 2. And I'll just point out that this picture on the right here, the brain is my pumpkin that I carved for our Renegade Research Halloween contest. Okay, so in the last meeting, we talked about the Brainstem. So we had Michael Van Elseker, Jeff Wood, and Braden Yelman. And Dr. Van Elseker went into details of the Brainstem and how there's sickness circuitry in the Brainstem. So like when you have an infection, the Brainstem is affected, and then it sends signals to the rest of the body, changing your body. It's evolutionarily, I just heard somebody, can somebody mute? So it's this evolutionary response so that when you're sick, it sort of slows you down, gives more energy to your immune system. And it can come from both, molecules in the body that are sending signals via a mere response across the blood-brain barrier, or it can come from signals through the vagus nerve, which can also be from non-infectious causes. So if you look at the Brainstem and everything that it can do, and then you compare to the systems and the symptoms in MECFS, there is a lot of overlap there. So the Brainstem controls things like the autonomic nervous system, the immune system, the GI system. It's related to sensory gating and the sleep and wakefulness. And within MECFS research, there have been abnormalities found in the Brainstem. And that's what Kiran will talk about today, some of their findings looking at MRIs of Brainstem in MECFS. So personally, I don't think that the mechanical issues cause all cases of MECFS, but I think it is possible that they can cause the symptoms for many people. So mechanical issues can cause deformation of the brainstem. And so if you look at the picture on the bottom left, this is an example of one of the mechanical issues, which is when, if you look on the bottom, this is a normal configuration of the spine. And it's possible in basilar invagination that part of the spine ends up actually compressing the brainstem. And there are other types of mechanical issues also that can cause different stresses in the brainstem. So for example, tension from if you have tethered cord, which is when the bottom of the spinal cord ends up getting stuck to the inside of the spine. And I mentioned it's possible that many people have these issues. We don't really know, what percentage overall has mechanical issues, but there have been many anecdotes of patients going into complete remission from having surgery or doing physical therapy for these conditions. And within the patient community, if you go on Twitter, there seems to be a lot of people that end up having these issues when they are actually investigated for them. But I will mention, not all people who have these treatments improve from it. So Dr. Yelman, that gave this the clinician's perspective of these conditions, went through how many of the conditions like CCI, craniocervical instability, and tethered cord and intracranial hypertension are diagnosed clinically and treated. And talked about how this can improve symptoms for many MECFS patients. But he also did mention, when he goes through a workup, this isn't necessarily what he investigates first. There are many other pieces that can help patients, like pacing and treating mast cell activation and other conditions. Okay, so that was my very, very high level summary of part 1. If you didn't see it, the recording of it is on our YouTube channel. So for part 2 today, here's the schedule. So we'll start off with Kiran, who will talk about some of their research in using MRIs and talk about some of the findings of brainstem abnormalities. And then Herbert will talk about one of his hypotheses and how it connects to the brainstem, how that might be involved. Each of those will be 20 minutes, and then we'll go to a 20-minute Q&A and discussion. And I want to mention, for this meeting, having the discussion is between all of the different people here is actually one of our main goals. So, in other meetings, it's more like the presenters are trying to teach the audience, and then it's mostly the audience asking questions of the presenters. But in this meeting, we encourage people to add information to the discussion that might not necessarily have even been presented. So we want to think of it more as like a conversation. So after that, we'll have a 10-minute break, which we didn't do last time, and we should have, because the meeting went over 2.5 hours. And then we'll go into some lightning talks. So these are very fast talks with one slide. The presenters will try to stick between like 2 and 4 minutes. We might be able to go up to, and then we'll go into more of that discussion. And we will kind of go as long as the conversation is still going and people still feel like they want to keep talking, which, like I mentioned last time, was like 2.5 hours. All right, I think that is it. Yeah, so let's get started. We'll have Kiran jump up here and share screen to get your slides up. Oh, I will introduce him too. So let me find my notes here. Okay, so Kiran is a research fellow at the National Center for Neuroimmunology and Emerging Diseases at Griffith University in Australia. He is using advanced multimodal MRI approach to study brain impairment in MECFS and long COVID. And his research focuses on understanding how these conditions affect the brain and to develop new neuroimaging methods to identify them earlier. So I will pass it to Kiran.
Hello, everyone. Thanks, Tex, for the introduction. Can you see my screen and hear properly? Can everyone listen?
Sounds
Yep.
good. You can see it.
Perfect. So yes, as said, I'm Dr. Kiran Thapaliya, working as a postdoctoral research fellow at Griffith University. So today I'm going to present some of our MRI findings, especially in the brainstem regions in MECFS. So this is the NCNED team at Griffith University, led by Professor Sonia Master-Gradisnik. Here we do multi-dimensional research, for example, immunology, MRI, and epidemiology. But today I'll be presenting some of our, again, findings, especially in MRI. I would like to thank every volunteers who have participated in our study, because it won't be possible without their support. So as we all know that MECFS patients suffer from neurocognitive impairment. They really struggle in processing speed. They have memory loss, attention deficit, and executive dysfunction. If this is the case, I mean, for the information processing and memory loss, brain is one of the key regions. And there are different regions in the brain, and one of the area that we are going to look is the brainstem. Why we choose the brainstem is that it is involved in the multiple function. For example, it is involved in the cortical arousal, consciousness, autonomic functions, attention and memory, problem solving, sleep-wake cycle, and motor control. If we look at the right-hand side, the brainstem is divided into 4 different regions: the midbrain, pons, medulla, and SCP on the right, which is superior cerebropeduncle. So these 4 regions makes the whole brain stem regions are involved in the multiple functions. So the main motivation of this study is to use multimodal MRI approach to study the brain stem dysfunction in MECFS condition. So we are, as I said earlier, we are going to use the MRI technique to study this. So whoever are not familiar with the MRI, it is MRI uses a strong magnetic field and radio waves to produce images. It's totally non-invasive method. We don't insert any contrast agent. And MRI is used to examine the anatomy and pathology of the brain activities, and changes in tissue structure, like tissue microstructure like myelin or iron. So there are different field strength available. For example, there are 1.5 Tesla, 3 Tesla, and 7 Tesla MRI. 1.5 and 3 Tesla MRI are mostly used for the clinical purpose, whereas 7 Tesla MRI are used for the research purpose. However, there are 7 Tesla MRI are available now for the clinical purpose also. So MRI produces different kind of images. For example, if you look at this on the left, it's the structural image which is used to study the anatomy. For example, the white matter, gray matter, and CSF, as well as to find the lesions. So if you look in the middle one, this is the example of functional imaging, which is based on the blood oxygen level that gives you the neuronal activities. And the right one side is the diffusion tensor imaging. This is a kind of MRI images that we can get from the MRI. And this is mostly used to study the axonal integrity, white matter, and even the myelin or myelin loss in the brain. So the 3 key points to remember, what I have presented now is that MECFS have neurocognitive impairment. Brainstem is one of the key brain regions involved in multiple functions. And MRI is used to study brain pathology using different MRI modalities. So now let's look at some of our findings using the MRI, especially in the brainstem region. In our first study, we use the structural images, and we acquire the data from 45 MECFS patients and 27 healthy control with 3 Tesla MRI. So here we have the two different images. That is the which is called the T1 OTET images and T2 OTET images. I don't want you to remember what is this, but what T1 gives you the brighter white matter, whereas T2 gives you the bit darker white matter. So we use some kind of image processing technique and calculate the ratio. That is called the T1 over T2 ratio, which indirectly indicates the level of myelin density in the brain. And this technique has been used in other studies, for example, in Alzheimer's, schizophrenia, and multiple sclerosis to identify the demyelination or changes in the myelination. So we use the similar technique for 45 MECFS patients and 27 healthy control. So what we found here is that the T1 over T2 signal intensity was significantly higher in MECFS patient compared to the healthy control. If you look at this white arrow, it shows that this signal intensity was significantly higher, especially in the brainstem region. So why does this happen? I mean, what does this indicate? This indicates that higher signal intensity could be potentially due to increase in myelin and iron content in MECFS. So the question might be like, why does this happen? This could happen because most of the white matter structure may undergo compensatory or neuroplastic changes during disease progression. And these compensatory changes may be related to an increase in myelin or iron in MECFS patients. So this is the another modalities called the functional MRI. So this is mostly used to study the brain activity during the resting state or the task-based state. So this is very important because it provides a deeper understanding of cognition and behavioral functions. It mainly works on tracking the blood flow and provides the most active regions in the brain. And with these modalities, we can measure the connectivity between the two brain regions and see which regions are weakly or strongly connected with one another. So we use this MRI technique to study the brain connectivity in MECFS patients. So mostly we use the task-based functional MRI. So this task is called the Stroop task. We choose this task because it needs some it needs more pretty much attention, and it's very cognitively challenged. So when we put the participant inside the scanner, this task appears inside the screen, and the task is like, does the color of the upper word corresponds with the meaning of the lower word? And if we look at this on the right hand side, we have got three different blocks, which is the congruent, incongruent, and neutral. So this is the blue one. The color is blue and it is the blue, so it's yes. But if you look at the middle one, it's very cognitively challenged because it's a bit confusing also. The color is green and the words is blue. So most of the participant gets press the yes button here, but this is actually no, because they read the word at the top and the bottom and they try to click yes, but it is no. So this is one is the green one. This is called the neutral one. This is also no. So this is called incongruent tasks, which requires more memory and attention. So we want to see, okay, what happens when the people perform the tasks? Is there weaker connectivity or the stronger connectivity in MECFS patient compared to the healthy control? So we acquire the functional data again from the 45 MECFS and 27 healthy control from the same cohort. So interestingly, we found that the connection within the brainstem, that is from the medulla to the cuneiform nucleus, that is the part of the midbrain, is significantly has weaker connectivity in MECFS patient compared to the healthy control. So there is some impaired connection within the brainstem region, and I think this could be because, when we have as I said, there is the brainstem is involved in the multiple function. So there is that's why like MECFS patients are suffering from sleep-wake cycle, memory, balance, all these problems are pretty much related, could be related to the brainstem dysfunction. So so out of 45 MECFS participants, we try to classify, subgroup them into the different case criteria. So I think people are pretty much familiar with the different diagnosis criteria for MECFS. It's not, they are mostly Fukuda, CCC, or ICC. So out of 45 MECFS patients, 24 met the Fukuda criteria, 18 met the ICC criteria and healthy control. As we know that the Fukuda criteria is pretty wider, has a broader symptoms, whereas ICC has a stringent criteria. So we would like to see, is there any connectivity difference between Fukuda versus healthy control and ICC versus healthy control. So when we did some experiment here, we found that there was a weaker functional connectivity between the medulla and cuneiform nuclei in MECFS patient meeting only ICC criteria. We did not find the same regions weaker connectivity in MECFS patients meeting Fukuda criteria when compared to the healthy control. So in our next project, in our next study, we use the diffusion tensor imaging. So I wouldn't like you to go to the understanding of details, but what the key point is, it provides you some information about the white matter integrity and the degree of myelination in the brain. And this mostly works on the in the diffusivity of the water molecules. So we use the same cohort. Again, like we want to see, okay, we have seen some differences in the functional connectivity in MECFS patient only meeting ICC criteria. So, and we said, okay, let's see what happens in using these imaging modalities. So we lost the 2 MECFS patients meeting Fukuda criteria because of the image quality, and we have the 18 MECFS and 26 healthy control, and we use the 3 Tesla MRI to acquire the data. So here again, we found the significant differences in the DTI parameters, which is diffusion tensor imaging, and the differences were only seen in MECFS patient meeting ICC criteria. So if you look at this figure here, this is called the MD, which is the mean diffusivity. This measures the degree of myelination. So if you have the higher MD or the mean diffusivity values, then that is the demyelination. If it's lower, then it is called there is some higher degree of myelination. So in MECFS patient, we found this MD value was significantly lower, which is relevant to the higher degree of myelination in the brainstem. So when we compare, there was no significant differences or found between MECFS patient meeting Fukuda criteria and a healthy control. This is actually in line with the MECFS diagnostic criteria, because Fukuda criteria does not necessarily need to include neurocognitive, cardiorespiratory, or thermoregulatory impairments now required in the ICC criteria. Therefore, we may need to use stringent criteria for the proper diagnosis of MECFS. So take-home message from this study is we use multimodal 3D MRI that showed brainstem impairment in MECFS. We also found weaker functional connectivity within the brainstem, and DTI showed altered tissue microstructure, for example, myelin in MECFS patients meeting only ICC criteria. So now we use the ultra high field MRI, that is the 7 Tesla MRI in our studies, and see, okay, can we see some more anatomical structure, or can we find the better images using the ultra high field MRI. So why we choose 7 Tesla MRI? Because it provides the better quality images, improved visibility of pathological alterations, a clear depiction of anatomical detail. If you look at the arrow here, you can see this is the left-hand side over here. This is the 3 Tesla MRI data, and this is on the right-hand side is the 7 Tesla MRI data. And you can see the white matter is very much brighter here in the 7 Tesla MRI as compared to the 3 Tesla MRI. So in our study, so we're gonna see, okay, what about the functional connectivity? Can we see any differences is the weaker or the stronger connectivity in MECFS patients when we compare to the healthy control. So we acquire the functional MRI data from using 7 Tesla MRI from 31 MECFS patients meeting ICC criteria and 15 healthy controls. So we use the same Stroop task again in the 7 Tesla MRI also. So we found that there was significantly weaker connectivity between brainstem and cerebellum regions in MECFS patients when compared to the healthy control. So as we know that cerebellum is one of the key brain regions, and it has got the largest number of neurons. And this cerebellum region is mainly involved in the maintaining balance and coordinating volunteer movements, for example, eye movements, motor learning, and cognitive functions. And these cerebellum regions communicate to the cortical region, that is the cerebrum, via pons. So if there is some impairment connection between the cerebrum and brainstem, multiple functions are affected. For example, cognition, attention, decision making, and even the balancing or the physical function. And we know that the physical function is very, very low in MECFS patient. So this could be the reason why that connection between these two regions is contributing to most of the symptoms experienced by MECFS. So we all know that COVID-19 arrived in early 2020, and people are still suffering from it. And people have shown that there is some brain impairment in COVID-19 people, or even after they are recovered from the COVID-19. And most of the COVID-19 people, at least the 10% of COVID-19 infected people, develop themselves into long COVID. And there has been like so many findings that long COVID has similar symptoms compared to MECFS. There is a symptoms overlap between the MECFS and long COVID. If that's the case, long COVID had the difficulties, again, the neurocognitive impairment. If that's the case, do they have any structural or functional changes in their brain? Let's find it out. So again, we use the 7 Tesla MRI. We use the functional MRI. This is the pilot study conducted in the 10 long COVID and 13 healthy controls, and all of the long COVID participants made the WHO criteria. So when we do the functional connectivity, similar to the previous one, but we found the functional connectivity within the brainstem region or strongly connected within the brainstem regions in long COVID patients compared to the healthy controls. Now, this is pretty contrast what we found in the MECFS. Because in MECFS we found the weaker connectivity, whereas here we are seeing the stronger connectivity. So this could be due to, because if we look at the MECFS patients, in our case, the disease duration were almost like more than 10 years. Or for the long COVID participants, the disease duration was almost 1.5 to 2 years. So that could be the one reason. And another reason is that histological experiment have shown that there is some RNA viruses, positive SARS-CoV RNA viruses, even in the brainstem region. And due to that, there might be some activation of microglia activations going on. That's why the brainstem has to have requires the more bold signals, that is the more blood level, to perform the tasks properly, or to perform the brain function. So another thing could be like in the brainstem also, like in MECFS patient, most of them meet the stringent criteria, like for example CC or ICC, whereas in long COVID these criteria or the stringent criteria were not met. But we never know. We might require a longitudinal study to find out whether the long COVID participant will have the similar brainstem dysfunction like MECFS. For example, they might all this stronger connectivity now could be turned into the weaker connectivity in the longer period of time. But that needs to be done find it out by the longitudinal study. So take home message, what we found here is that 7 Tesla MRI reveals distinct brain connectivity patterns in MECFS and long COVID, potentially explaining their shared and unique symptoms. So as there have been some literature showing the overlapping symptoms between the MECFS and long COVID. So we know that there have been some literature showing like at least 13 to 58%. The variance is pretty big, but at least like I would say the 13 to 15% people of long COVID patients meet the MECFS criteria. So if they meet this criteria, do MECFS and long COVID patients have similar brain alteration? So what about that? Because they have, both of them have the neurocognitive or the brain fog. If that's the case, what about the brain alteration? Are they similar or the difference? Let's find it out. So we acquire the structural images from 7 Tesla MRI. So we have 10 MECFS participants at long COVID and 10 healthy control. So what we did is we estimated the volume of midbrain, pons, medulla, and superior cerebral peduncle, and see whether they were significantly different comparing to each group. So here, I would say the red bar is MECFS, the green is long COVID, and the blue is healthy control. The y-axis is the volume. We can see that the MECFS and long COVID, the pons volume of MECFS and long COVID were significantly larger compared to the healthy controls blue. Similarly, the whole brainstem region was also significantly larger in MECFS and long COVID when compared with healthy controls. And similarly with the superior cerebral peduncle, which is SCP. So again, we found the significantly larger only in the long COVID participants compared to the healthy controls. So this could be like, the increase in this volume may reflect to the edema or inflammatory responses, neurodegenerations, or viral invasions, which has been again shown by the histological experiment. So there were no significant differences in the brainstem volume between the MECFS and long COVID. This is again like which is very consistent with the other findings. We show that there is some kind of overlapping presentation between two conditions. It also shows that there is some link between MECFS and long COVID. So we did the correlation, pons volume and correlations with the part of the correlation with the pain measures in MECFS. Here the pain was measured from 0 to 100. 100 means no pain and 0 is the extreme pain level. So we found that when you have the severity, when you have the extreme pain level, the pons volume actually decreases. This could be because the brainstem regions have several nuclei that receive ascending and descending signal pathways that inhibit or facilitate pain by upward or downward regulation of neurotransmission. So actually the pons have a reticular activation system nuclei that inhibits or facilitates the pain in the brainstem, within the brainstem, or from at the top or the top of the cortical regions or to the spinal cord. So the take-home message is that shared brainstem abnormalities in MECFS and long COVID suggest a potential common underlying mechanism. So I would like to conclude my study is that we showed that multimodal 3T and 7T MRI had showed brainstem abnormalities in MECFS and long COVID, impaired functional connectivity, alteration in tissue microstructure, and brainstem volume were found in both conditions. MECFS and long COVID have similar brain alteration, suggesting a link between these two conditions. So what next? The NCNED is conducting longitudinal study to identify progressive brain tissue changes in MECFS. We are also doing multi-site study using same case definition, especially CC or ICC, which is Canadian consensus criteria or international consensus criteria. We are also running clinical trials to find the treatment of MECFS and long COVID. And we are studying MECFS and long COVID together because their symptoms overlap. So I would like to acknowledge or thank funding bodies like ME Research UK, Stafford Medical Research Foundation, National Health Medical Research Council, and all the national and international collaborators. Thank you.
Thank you. It is super interesting. So we're going to go straight into Herbert's talk, and then we'll start the Q&A after that. I'll mention that the text Q&A is open, so you can go in there and submit questions in that, and people can upvote, and then we'll bring up some of the people who got the upvotes for their questions during the discussion. So Herbert, do you want to share your slides now?
Sure, will.
Oh, and while you're doing that, I will introduce you. So Herbert is a pediatrician researcher and MECFS patient. He published on the possible role of neuroglia in MECFS, together with Marie-Eve Tremblay and Dorothy Bendy, and presented a hypothesis on BCG vaccination for MECFS and long COVID. He's affiliated with the Health Services Research Group at the Department for General Pediatrics, Pediatric Cardiology, and Neonatology at University Hospital in Germany. All right, go ahead.
So, thanks, Tess. Well, I called it Guardian Angels Off Duty. After the talk, you will realize what that's all about. I must say, thank you, Kiran. See, I'm neither a brain nor a brainstem expert. I consider myself somewhat familiar with MECFS as both patient and researcher. Well, unless you have it, MECFS is actually a fascinating disease. I think most fascinating is actually the clinical core feature of post-exertional malaise. Basically, that we have symptom exacerbation after many, many different modality stressors, be they physical, cognitive, mental, orthostatic, emotional, social, sensory, or chronobiological. So this is really, whenever there's stress overload on an individual level, with a very individual threshold, you will have the hell of symptoms. Yeah. So the other feature is the bewildering pathobiological basis. Well, see, there is, you're all familiar with that. There is so many disparate on the one hand, but on the other hand, they may all fit together. Pathobiological features, including immune dysfunction, inflammation, reactivation of microbes, etc., mitochondrial dysfunction. We all heard about that many times. The only problem that we have is we don't know how the arrows run. So we don't know, what's primary, what's secondary, what's upstream, what's downstream, what's a bystander phenomenon, and what's really driving the symptoms, so to speak. So it's a very, very complicated, tricky picture. Now, I think maybe post-exertional malaise may be actually a good chance to disentangle the whole mess, because indeed, MECFS, different from fibromyalgia or even MECFS by Fukuda criteria, comes in two different clinical stages that every patient is very aware of. You have your normal days, baseline days, and then you have your bad days, post-exertional malaise. So really, it's two different diseases in one, so to speak, or two different, not diseases, but different stages in one. And I really believe that if you understand PEM, you understand MECFS. Because looking back, we've had a lot of problems with not really considering this two-faced quality of the disease. So there are like hundreds of published studies on MECFS, which basically harvest their samples, their data, both from good days and during bad days. So we put them all together in one pot, and then we analyze them, and then nothing really significant appears. There's a lot of signals, but a lot of noise. And it's very hard to tell. We had this whole gamut of problems with cytokine research. One study saying HIP, one saying HUP, one saying this cytokine goes up, one another. So no clear picture, but I really am not really surprised, because if you pick samples from two different stages of the same disease, then you really should not be surprised if you have, like what I have here on the slide, kind of a fruit that doesn't make sense. Yeah. So I think as we get better now in defining this disease, as a matter of fact, we up to now, after like 50 years of research in MECFS, we don't have a phenomenological, phenotypical description of the disease itself, because we have no single cohort of patients that has ever been systematically followed longitudinally. You have to imagine, we have a very common disease where we don't know much about because we have never studied it longitudinally. We now have first case reports, where I'm very happy about, of single patients studied and analyzed during baseline and then during disease exacerbation. We also have imaging studies during that, but I will refer to. But we have a great lag in a clear definition of the disease. I still hope we once have in the next future, we have like I would call it the hero study, like take 100 MECFS patients and follow them on good days and bad days. Just draw labs like the heck out of them on good days and bad days, and then compare. And you'll have signals. You will not have noise. You will have signals. I fear that we have the same problem now in long COVID research. I hear a lot about there are microclots. Yeah, there are microclots, but have we ever looked at if they are like present every day or during symptom exacerbation? Do they change in level? I mean, if they drive the symptomatology, you would expect that they should be different in abundance between good and bad days. We don't know. We have studies out of Germany, like red blood cell stiffness is increased in long COVID. Well, that's interesting, but now let's look, is that present all the time? Or is this only present when the patient feels really sick, so during bad days? And that's the privilege of the moderately affected that we can study in this respect. But these these dark days are finally coming to an end, I think. Here comes the sun. This comes out of exercise studies, because there we basically tweak the system by having the patients like study them during baseline and then stress them out, give them PEM basically, and then study again after 24 hours, 48 hours, whatever. Well, and the sun comes, it's all right. The results seem to converge on two very interesting findings. Finding one, at least in one-time efforts, MECFS patients can exert themselves, at least so, yeah? So this is surprising if you see how sick you feel sometimes, that you can, if you do it one time, you can exert yourself, at least as a moderate patient. But then comes the bummer. In the healthy controls, exercise induces a flurry of adaptive changes on many, many biological levels. Yeah, there are metabolic changes, there are transcriptomic changes, T cells are altered, natural killer cells are altered, monocytes altered in functionality, extracellular vehicles in abundance. You see these different results in the blood, you see it in the CFS, you see it everywhere. Now, what happens in MECFS after exercise compared to baseline? Nothing. Compared to healthy controls, MECFS patients show lack of metabolic adaptation in response to exercise, lack of transcriptomic adaptation in response to exercise, lack of changes in circular RNA expression after exercise, lack of adaptation of T cells and natural killer cells in response to exertion, lack of the normal rise of the concentration of extracellular vehicles after exercise. So really, there's basically no significant adaptive response. And so it goes on and on. There's a lack of changes in microRNA levels in the peripheral blood and blood mononuclear cells, lack of adaptive response of classical monocytes, lack of the acute phase response to luminal gut antigens translocated into the bloodstream during exercise. That happens during exercise, that we have a microbial challenge. Microbes get translocated. There is no acute phase response. There is also a lack of adequate rise of cell-free DNA in the blood after exercise, which is typical for exercise. In exercise, that really goes up. And so no surprise, there's a lack of cardiovascular and respiratory adjustment in response to exercise. So this is really significant findings. I have one example here looking at the urine, what changes in the metabolome of the urine with healthy controls compared to MECFS patients. Well, if you take the metabolome and you see only a few differences between patients and healthy controls before exercise, so that's kind of surprising. There's not much of a difference in the baseline differences before exercise. Now, what about 24 hours later after exercise? In the healthy controls, a lot of compounds significantly alter their abundance. You have the graph here, how much they alter. 255 compounds are significantly different from baseline, and all but 5 are increased in abundance. So we have 250 metabolites that increase in abundance after exercise in healthy people. Now, what about in MECFS patients? Not one single compound. There is no change from baseline. Post-exercise, urine metabolome looks exactly the same as pre-exercise. So here is the adaptive response to stress in MECFS patients. Total flatline. Now, what about the brain? Kiran, hope you're here and with me. First of all, there's a lack of adjustment of cerebral blood flow in response to orthostatic stress. Well, we know that. And this reflects a brain problem, not a cardiovascular one, because the cardiovascular indices do not correlate with the changes, but the vasodilation within the brain does. There is a lack of oxygen level adaptations in the brain after cognitive exercise. So you challenge these patients cognitively, and they don't change in oxygen concentration where they should. There is also a lack of adjustment of the resting state network in response to physical exercise. So the default mode network, which basically governs our resting activities, chill mode, resting, then the default mode network is activated, it's on. And the default mode network actually remains activated after exercise. So the opposite as it should. So instead of adjusting, the body remains in the chill mode. Now you could say, well, this is interesting, but really this is a problem. This is a huge problem. And to really realize what kind of a problem this is, we need to shortly revisit the stress response. The stress response physiologically has basically 2 arms. The first arm gets the body ready for action. Yeah, release the troops. You have that young boxer lady there, and that's the adrenergic surge, fight and flight, and etc. So get ready for action. The second part adapts the body to the noxious effects of exertion. It releases the guardian angels. So I say the noxious effects of exertion, yeah. What we underappreciate is that exertion of whatever level, in fact, represents a noxious assault on the body. There's so much that happens. There's inflammatory assault. I read a study on marathon runners. Their interleukin-6, so inflammatory signaling goes up by more than 100-fold. Yeah, so there's the body is raging. There's inflammatory assault. There's oxidative assault. There's adrenergic assault. There's metabolic assault. Metabolites change. There's mechanical assault, microbial assault. All kinds of noxious things that need to be regulated. They need to be to find an adaptive response. So interesting, while the first arm of the stress response seems to work okay, at least in the moderately affected patient, the second arm, the guardian angels, seem to be lacking. So post-exertional malaise to me very much looks like we patients get injured by friendly fire. We should be protected. We exert ourselves. Yes, we can. The first part of the stress response works just fine, but the guardian angels are missing. So exertion hits the body unbuffered. And what can happen? I already mentioned, there are quite a few things. Inflammation, viral reactivation, mitochondrial dysfunction, immune dysfunction, endothelial dysfunction. Just imagine the onslaught of oxidative stress, what that can do to your immune cells. They will stop working properly if they remain unbuffered. Endothelial dysfunction, metabolic dysfunction, and so on, neuroinflammation. So basically what can happen after exercise, if it's unbuffered, the whole zoo of ME/CFS. All these things in this graph basically can, by the textbook of physiology, happen if the stress response hits you unprepared. Including some vicious cycles, of course. See, oxidative stress can cause the endothelial cells to malfunction. That will make for a leaky blood-brain barrier, for example, or reduced cerebral blood flow, neuroinflammation, and so on. Or you can have viral reactivation in endothelial cells. What we sometimes don't really appreciate is that some of the endogenous viruses, like some enteroviruses and also herpes viruses, actually lodge in the endothelial cells. So imagine if we get a load of inflammation onto the endothelial cells. These may flare up, and we still don't know if endothelial dysfunction may be actually related to a flare-up of a latent microbial load. You can also, from endothelial dysfunction, of course, you will get reduced tissue perfusion. You will have mitochondrial dysfunction, which means the mitochondria will start to use alternative fuel like dirty fuel, amino acids, and so on. That will produce toxic metabolites like ammonia, which will then cause neuroinflammation. You know, ammonia is a very powerful stimulant of astrocytes. Actually, they swell up and they make really bad neuroinflammation. Plus, of course, an inadequate stress response. So you have all these vicious circles that can roll, that we patients actually feel. Yeah, they feel them rolling. Now, the talk was about Brainstem, yeah, well, Brainstem. So you may wonder, what does this all have to do with the Brainstem? I'll get to it. So the stress response, looking at from the brain level, is the result of intense collaboration between certain CNS regions and nuclei, and the functional networks to which these are connected. So a coordinated stress response actually requires normal connectivity, communication between networks, between centers, nuclei, regions, and also between functional networks. And it requires a normal hardware, so the nuclei themselves. The CNS regions and nuclei implicated in arousal and stress adaptation, Kiran will know much better than me, I may later give a comment to that, include many of the regions that we talk about in ME/CFS. Actually, it's like you go through the textbook and then you notice, oh, that's all the research about what pops up. The hippocampus, amygdala, subdivisions of the prefrontal cortex, cingulate, including parts of the cingulate cortex, insula, thalamic regions, and the circumventricular organs, which seem to play a specific role in stress regulation. I didn't know that, but I read that those being very much unprotected, or by a rather leaky or rather easily to overcome a blood-brain barrier, do play a significant role. And then, of course, the brainstem. I think we have heard about Brainstem enough to understand that it's a really central and has a coordinating role in the stress response. Big surprise, a recent study that just came out a couple months ago shows that the brainstem is a part of a functional connectome linking 58 brainstem nuclei with the midbrain, the pons, the medulla, and the cortex, probably the cerebellum too, if you measure. So yes, the brain findings identified in MECFS basically point to many of those brain hubs, nuclei, and networks relevant to the stress response. We do see broken connections, which my term for altered connectivity, between the brainstem, the cerebellum, brainstem and the cerebellum, and the brainstem and higher nuclei relevant to stress adaptation. We also see abnormal perfusion, oxygen content, and metabolic function in several brain regions relevant to the stress response. For example, the hypothalamus, the cingulate cortex, insula. And we had this talk before, abnormalities in the brainstem itself. We don't know what they come from. Maybe inflammation, maybe vagal dysfunction. We don't know. We've heard from in the intramural study that there may be reduced norepinephrine signaling going on in the brainstem, so that it can't activate the prefrontal cortex or other regions. We don't know. There's something related to the brainstem, and that actually makes a lot of sense from all that we heard. Again, brain connectivity, of course, is heavily influenced by brainstem nuclei. And also the brainstem is a recognized regulator of our peripheral biology. We recently had this finding even of a master switch of the inflammatory systemic response. So if that master switch doesn't work right, then the peripheral inflammatory response is augmented. Then the brainstem, of course, is the relay station picking up all the signals from the body. So basically, you can consider it basically a part of the periphery. Yeah? So it's a relay station. Then, of course, one argument is it's shown to be affected in MECFS. And of course, the central role of the brainstem would also explain the clinical and pathobiological overlap between MECFS and traumatic brain injury. I did a review 3 months ago for an interview of the overlap between traumatic brain injury and MECFS. Well, it's striking. It's not only striking in clinical overlap. So, they have this fatigue. They may also have this exercise tolerance. It's not well described, but there's also some indication that they do have this problems with overexerting themselves. They have sensory overload issues. They have this brain hyper-excitability. They have the sleeping issues. Go through the Canadian consensus criteria, and you basically hit traumatic brain injury. And the same also applies to the pathobiology. There is so much going on there. There's endothelial dysfunction. There's immune dysfunction. They have so many things in common that, of course, we should basically also see this as an indicator that the brainstem being affected in MECFS. And of course, it could explain the mechanical assaults that they can also cause MECFS by twisting, torturing the brain stem. We heard about this last time. I also agree that there may be some cases of purely mechanical etiology. I think that most cases of MECFS are actually related to immune overload. It's not necessarily only infections. It can also be other immune relevant stressors that overcharge, overtax the system. Now, does the abnormal stress response that I talked about explain MECFS? Well, I don't know. This is just a hypothesis. I clearly think we can observe this, but I don't know how it fits into the bigger picture. Because of course, MECFS not only supports a disrupted stress response, but also other CNS dysfunctions like autonomous dysfunction that may or may not be related. But I do think that the abnormal stress response may explain PEM, and that's not a trivial thing. It's not trivial because we have so many studies that we do, and that we should never forget. Are we really talking about MECFS, or are we talking about something different? See, we have those, for example, IgG transfer models. Those are super exciting. Yeah, we take blood or serum from a CFS patient or a long COVID patient and put that into a mouse, and the mouse comes down with fatigue and MECF-like symptoms. But is this MECFS? Well, MECFS in its core has this one feature, and that's post-exertional malaise. So as long as we don't really show this, we have to say, well, we transfer something with the same experiments are positive in fibromyalgia. I'm not surprised, but we always have to keep in mind, never forget to ask the PEM question in research. When you do research about MECFS, well, then you need to answer the questions around post-exertional malaise. Last question. Questions. What could be the root cause of all this? This is pure speculation. This is just taking leads from studies published, and interestingly, many have been published in the last 2 years. It was shown that infections actually can induce connectivity changes in the brain. Even a mild SARS-CoV-2 infection can lead to persistently impaired neurogenesis in certain brain regions, also in decreased activity of oligodendrocytes and myelin loss. So actually in dysfunctional connectivity. So this could be the starting point, basically, that there is some disrupted, broken connections, to speak, that disrupted connectivity, which then sets the stage for all these vicious cycles going on. Also, very interesting, recently published, infections can actually induce a priming, a kind of priming or sensitization of the CNS against future inflammation. And that was shown for SARS-CoV-2 antigens too. So it floods the brain, that's how I think about it, and leaves it hyper vigilant, sensitized against future inflammations. And now think what happens. Future inflammations could also include viral reactivations. We know that the immune systems over time will become exhausted, and we have more and more viral reactivation after overexertion, for example, and that could well be those inflammatory triggers that then are will have a greater response due to this priming and sensitization of the CNS with the original infection. And of course, here the finding of this master switch in the brainstem comes to my mind. If that master switch that basically regulates systemic inflammatory response, if that is becoming more sensitive, then that could mean that with every new viral reactivation, those patients just stay in their in the red zone. Infection could induce inflammation of the vagus nerve. We've talked about this quite many years back already. Could also induce autoimmunity. That was recently published too. They found they found 3 novel targets in long COVID patients with autoimmune targets with targets in the brain. Now imagine these targets were, let's say, on astrocytes, so neurovascular coupling would go bust. So the brain regions wouldn't be perfused right. That could be a starting point. So we will, of course, in the next future pursue these leads further on, but I think this is quite interesting because, well, autoimmunity is always a question how much of MECFS relates to autoimmunity. It certainly is not directly related with post-exertional malaise because autoantibodies have a long half-life and they are not produced just on the switch. So I doubt it, but autoimmunity can well play a role by priming, for example, the CNS to respond differently and to have a messed up stress response. Then, of course, the classical hypothesis that infection could induce endothelial dysfunction, which we see in COVID, and then lead to a leaky blood-brain barrier, and then or even dysfunctional neurovascular coupling, neuroinflammation, and thus dysfunctional connectivity. So we have quite a few models which could explain how the system, how the CNS gets primed and gets started to have a messed up inappropriate, inadequate stress response that then sustains and perpetuates the whole problem, yeah, with ongoing viral reactivation, with ongoing metabolic changes, with ongoing mitochondrial dysfunction. So that would be my summary of my take on MECFS, especially of post-exertional malaise. If you want to read on, I put that on the internet, Guardian Angels Off Duty. So now you know what I mean with Guardian Angels. I wish everyone good Guardian Angels for the future. Thank you.
Thank you. Yeah, we got a comment in our panelist chat that we really have to be asking the PEM question. So I think this is super interesting for you to have something that could possibly explain it. We also have a clarification. I just want to make sure that everybody knows that when you're talking about stress, it's a physiological process, not necessarily life and emotional stress. Just want to make that clear for the public. Like lifting weights would be stressing, exercise. So just wanted to make sure that people know that. So let's see here. I want to remind everybody that you can go into the Q&A and upvote the questions that people have been putting. And while you're doing that, we'll start to take some questions or some discussion from the panelists who are already up here. So if you want to raise your virtual hand, if you want to come up on camera. Jeffrey.
Yeah, thank you. Jeff Lubell, I'm the parent of a child with MECFS. And I wanted a comment is for Dr. Renz-Polster. So I really appreciate your perspective that we need to adopt a longitudinal lens and really look at whether patients, where they are in the cycle of PEM and non-PEM. But I'm curious about why you limit your perspective on longitudinal analysis to simply that one cycle. It seems to me that there's a much longer term longitudinal lens that we need to take and consider that patients may be at different points in their recovery journey. They may be getting better, they may be getting worse, and that better or worse may be corresponding to changes that are related to the pathophysiology of the illness. So just to give a quick context, my daughter has had 2 major relapse events that caused her to become substantially worse. And in between, she's had periods of gradual recovery, excruciatingly slow recovery, but recovery. And her response to exercise at the beginning of that recovery cycle is quite different than it was at the end. And if we continue to focus all of our energy on cross-sectional research without distinguishing between people who might be at the beginning or toward the end of a recovery cycle, we're never going to understand this illness. So I'm curious about whether you would endorse a broader view of the importance of longitudinal research.
Absolutely. I mean, if do you want me to comment right away? I think that would be perfect. Of course, the immediate longitudinal changes, like in the boom crash cycle, those are very interesting. But of course, the longer run of the disease from a better shape to worse shape, or vice versa, of course, and the vulnerability that this entails. Sometimes you get more vulnerable, as you said, to PEM, so your threshold goes down. And that would be super, yeah. That my hero cohort of 100 patients should not just do a boom bust cycle. In terms of PEM, it should also, of course, follow them during a long stretch.
Right. Thanks.
Yeah. Bo?
So can you hear me now?
Yes.
Are you hearing my voice?
Yes.
Okay, wonderful. Well, I really appreciate these wonderful presentations of Kiran and Herbert. And I agree with Herbert's entangling what is really the root cause. And I dare to raise my hand as I did 25 years back in a similar situation where we were actually on the main question was on fibromyalgia, but in our cohort here in Sweden, we actually, when we look back and if we look at the criteria for fibromyalgia, a very large portion of our patients with ME, according to the Canadian criteria, also have fibromyalgia. Its overlap is very, very high. I think we had in our 220 or 231st patients, we had an overlap of over 80% between fibromyalgia and ME. And as I said, 25 years back, I raised my hand in a large international gathering, and I said, what about if fibromyalgia is not a disease in itself, but it's a matter of flow in the craniocervical area? And I still remember that situation because there were thousands of colleagues, and I felt like they all looked at me like an oof or stupid person. But after that session, it was on it was the whole situation was stopped by the attack on the. But afterward, anyway, there was a neurosurgeon, Dan Heffes from Chicago, who approached me and said, "Well, that's an interesting thought." And then he told of how he had made surgery on a then 36 patients, I think, with spinal canal obstructions. And he said, I was surprised because I operated them for spinal stenosis in the cervical spine area. But afterward, when I interviewed them, they told that they had been freed from their fibromyalgia. And he published this study, and he also has published another one in 2008, I think. This was 2001. And I think some of you may have read our article from a few years back now, where we actually looked on the MRIs of the brain and the cervical spine on our patients, also the 221st. Now we have 2,000, but and it was surprisingly high prevalence of obstructions, different kind of obstructions. And what I'm asking is, Herbert, you in your final discussion here, you point very much to the infectional possibility. What are your thoughts on what about if one of the main basic things would be a flow thing? Flow of the cerebrospinal fluid and the maybe even the blood flow in this area.
Yeah, but one would refer to etiology and the other would refer to the pathobiological basis. So one wouldn't exclude the other. That's would be my comment. So any immune relevant event may bring on pathobiological changes that will then induce secondarily or whatever, or could induce other changes. For example, you could imagine you have a disrupted blood-brain barrier. You have what we can also see, a disrupted neurovascular coupling. What that means for the flow of cerebrospinal fluid, I don't know, because I'm not a neuro guy, but I can imagine that all this sets the system up for like fluid leaks We do see, or we had seen these punctured hyperdensities that may correspond to like fibrin or fibrinogen being transvasated into the brain, which then could also easily change the circulation and the flow of the spinal fluid. So it's hard to disentangle probably what comes first. So if there is inflammation, you can easily understand, or if there is also if there is immune dysfunction. The immune dysfunction basically makes your glial networks not work as suggested, and this can end up with problems of CFS flow. I could imagine. So we would have to ask the neuroscientists.
Yeah.
All right. Quick clarifying question for Kiran from the Q&A. Did MECFS have more or less myelination? Audio was not clear. Oh, we can't hear you.
Can you? No.
Oh yeah, can you get a little bit closer and then maybe?
Okay.
There we go.
Yeah. Can you hear me now?
Yeah.
Yeah, perfect. Yeah, so it's very hard to say it's a high myelin or the low myelin. But what we have found from our study is that it's some kind of changes in the degree of myelination. So what we have seen is there could be potentially, I would say relative, it's not a direct measure, it's an indirect measure. So, but what we have shown is that some kind of changes in the degree of myelination that could be an increase in the myelination.
All right, thank you. Let's see. So we're running a little bit over time, but so I think we'll take a little break here like we planned for the patients. And then we'll come back and do a few lightning talks, and then we can continue this discussion with the people who are planning to ask questions. All right, so let's come back at, let's say 12 or 2:25.
Okay, great.
But I'll say, let's see here. I did grab a quick bio for you. So most important thing about John is that he's our Renegade Research Medical Director. And he's has 30 plus years in patient care, workflow tech, healthcare research, clinical informatics, and geriatric rehab, and also has a background in electrical engineering. Go ahead, John.
Great. And just double checking, are you seeing a slide from side to side?
Yeah.
Okay. So what I tried to do with this few minutes here is trying to tie together what happens at the micro level and the macro level. And a potential option is that triggers, whether environmental or infective or physical, such as a head injury, end up triggering the beginnings of injury and disruption. And then you have, when you get injury, you have the breakdown phase, the M1 white blood cell phase, and then you have the build phase, the M2 builder phase. And when that happens, you can end up at a micro level with hypoxia. And if you, so for example, in long COVID, with microclots blocking capillaries, you could end up with hypoxia. In a stroke without, with a different clot causing a macroclot, you end up with areas of hypoxia. When that happens, hypoxia itself will drive ongoing inflammation through what's called HIF1. And the thought is that once you have inflammation, then the hypoxia can drive ongoing inflammation. And some people are at a lighter level of severity and some are at a heavier. And then it could, in the skull, it could get to the place where that inflammation, because you're in a closed space, is causing more pressure and increase in pressure. And maybe somebody's predisposed to pressure. They have a small outflow for cerebral spinal fluid or something else. But either way, you're increasing pressure. And then if that pressure gets to the place where the brainstem is compressed, then is that worsening, or is that even MECFS from the perspective, and maybe the marker is this response to exercise and PEM that we were just hearing about. And so the 4 steps of it then would be, you have a trigger, whether environmental or infective, the injury, and we've seen, long COVID, you got millions of people around the world that way, and it drives inflammation. And this core piece, once you get inflammation, it starts driving itself because of the hypoxic tissue. And with this thing called HIF-1, when it goes into the nucleus, it spits out inflammatory constituents, and importantly, it spits out things that grow blood vessels, the VEGF-1. And so there's a yin and yang between HIF-1 and something called NRF2. And for example, a number of MS drugs are to augment NRF2, and when that happens, it cuts out HIF-1. But the bottom line is, in a closed space, that inflammation can get to the place where it could compress the brainstem, and we're seeing all the things we're seeing. So to help reverse it, it would be getting oxygen into that micro space. And really what you're trying to do is to help the body switch from the killer to the builder cells. And if you cut yourself, skin your knee, you see killer cells. Initially, stuff gets taken away, and then you see builder cells. Then you start forming a scab and starts cleaning up. And so an approach here at a system level or a person level could be quiet inflammation, break the microclots if they're there, support the mitochondria and gut, address MCAS, because in the interstitial tissues, you get mast cell release that happens when things get inflamed. That's consistent with the hypoxia. And then when it's all said and done and quieting down, limit the senescent cells that are around, the sort of zombie cells that are hanging out and not really replicating. And then, of course, address mechanical issues. And an example could be pacing to stay within the energy boundary so you're not creating more inflammation. And then things like curcumin and metformin work on the toll-like receptors and do some things with autophagy and apoptosis that might be helpful. The nattokinase has been shown in a number of places to be helpful with breaking down microclots. Probiotics can move the microbiome to short-chain fatty acids. Antihistamines, I think a lot are familiar with. Ficitin as senolytic that limits senescent cells was one of the drugs that helped, for example, in the Mayo long COVID clinics. A majority of people who on Ficitin had helped, and then trials with collars or other physical release. But the bottom line is multi-layered treatments addressing microinflammation and macro pressure to try and improve health. And it's analogous to what happened in cancer treatment decades ago, when you went from being, leukemia went from being 95% fatal for young children to basically the same amount curable. It was when people took some of the existing interventions and layered them in a way where their force of damage control was better, and their force of going after the disease, the root of the disease, was better. But rather than doing one at a time. And that's my talk.
All right, thank you, John. Athena, do you want to try and get your slide again? And we'll save questions until after these lightning talks too.
Can you see me? I can't see you, so I'm having some trouble here today. Can you see my slide?
We can.
We
yeah.
can.
Okay, great. Just one second. I'm just trying to see if I can see you here.
While you're doing that, I'll do a quick intro. So Athena is a CPA from Canada with CCI. Discovered what patients were reporting was in fact true, that diagnosis, imaging, treatment, and guidance were non-existent in Ontario. They've guided patients to upright MRI and treatment options, built a CCI-CCJ webinar recently, made available to the public by Dr. Ross.
Thank you for having me. I had a particular interest in linking the commonalities between patient groups. So Dr. Bowe, I'll call him Dr. Bob because I don't have a good affinity for names. Dr. Bowe spoke about some of the HEFS studies in 2004, Dr. Braggie 2020. And then we also know that recently Dr. Rowe has done some individual case studies in 2018 and started to longitudinally follow patient case studies dealing with stenosis and resolution through various complex craniocervical obstructions that have been linked. So that's where my passion lies. Today, my 2-minute topic is going to be the relevance of the craniocervical junction in the atlas axis alignment and the contribution towards maintaining optimal brain health with reference to venous congestion and MCAS inflammation. So it's a good follow-up to the previous talks. CCJ malalignment of various types we know can manifest in different outcomes and may contribute to neural compromise, cerebrospinal fluid obstruction, as well as arterial venous compromise. When mechanical compression of the jugular vein exists, obstruction of outgoing venous blood flow may be an issue, and cerebral spinal venous insufficiency has been observed in some neurodegenerative brain diseases. This is a pretty new conversation we're having. This past year, Dr. Huey formed a neuro committee comprised of neuroradiologists, neurosurgeons, neurologists, and the like. And some of the discussions included venous congestion and the overlap with IIH, idiopathic intracranial hypertension. Dr. Higgins describes venous congestion as the thing that no doctor bothers to look for. Also discussed is the high correlation between CCI patients and jugular venous compression. Although their surgical approaches are not consistently agreed upon, this is still in early discussions because really the cases are complex. They do agree that structural integrity of the CCJ is relevant in the decision-making process. And Dr. Huey suggests they know more than they did a year ago, and future publications are to be expected. What I've learned as a patient is that limited consensus for diagnosis and treatment options, whether invasive neurosurgical stem cell or non-invasive treatments are an ongoing issue, but doctors are doing their best to gain ground quickly. Many of us as patients around the world don't have access to neurosurgeons or fusion as a potential solution. For some of us, our efforts are strategically focused on mitigating persistent misalignment for as long a duration as possible and doing our best to ensure our lymphatic systems working at optimal levels with respect to the removal of metabolic waste. For example, atlas alignment obtained through upright MRI using accurate vectors has been a useful tool for some of us. I'd also like to add that patients with biomechanical issues experience a neuroinflammatory piece that rules our world. We recognize that there's an upstream biomechanical issue driving inflammation, which Dr. Rowe, Dr. Afrin, and others have recently started to integrate in presentations. Weaving inflammatory management solutions, which have been referred to by Dr. Houghton, such as mast cell stabilizers, have been very helpful in our attempts to rebuild CCJ integrity, some of us with remarkable results starting at ground zero. But I don't believe this data is being tracked, so I think there's lots of room to do so. Going forward, I'd like to suggest that a global consortium is a reasonable ask. This could be continued in the context of what we're doing and other forums, including experts and patient collaboration, where clinical outcomes and data are shared and the utility of tools have worked for them. There's lots going on around the globe, but limited sharing of information and lots of different non-consensus on different procedures that are being used currently. Thank you.
Thanks, Athena. Jeffrey, do you want to go next? Bring up your slide.
Sure. Sure.
And yeah, well, you're bringing that up. I keep forgetting the intros. Jeff is the parent and caretaker of a young adult with hypermobile Ehlers-Danlos syndrome, MECFS, POTS, cradiocervical instability, Chiari, tethered cord, and suspected mast cell activation. His daughter has been sick for 7 years. In his day job, he is a social science researcher. In his spare time, he is conducting research on her conditions.
Great. Thank you for having me. Really appreciate the opportunity to present, and I'm looking forward to learning more about Renegade Research and participating in the future. So it may not be surprising, given my earlier question, that I take a longitudinal lens here. I really want to make 2 points. The first is I want to offer some criteria for evaluating spinal hypotheses. And the second is I want to offer a particular hypothesis that aligns with these criteria. So, and this is really building on the talks that were made last time that I didn't attend, but I had the opportunity to read about. So first of all, I think it's important that we need to explain why people with spinal conditions are largely asymptomatic prior to falling ill. Right, so it's not enough to say someone has CCI, someone has Chiari, someone has tethered cord, but why are they not symptomatic until something happens, right? and to understand sort of the pathogenesis and the pathophysiology, we need to be able to answer that question. The second is, why do they become symptomatic once the illness triggering event happens? And then third, why does spinal surgery help to alleviate the condition? And I completely understand it doesn't work for everybody. I think it's highly risky. It's not my preferred option. We've deferred some of those surgeries. My daughter's had tethered cord surgery, but not CCI or Chiari surgery because of those risks. But I do think you need to be able to explain the pathway in order to have a valid hypothesis. So that's my criteria. Now here's my hypothesis, and it's actually very closely related to what Dr. Houghton offered, and you'll recognize pieces of this from different parts of the research. So from left to right, the first point is that you have a trigger, whether it's a virus or another trigger, that produces inflammation, endothelial dysfunction, and vascular damage, all of which increase vascular permeability. There's a lot of focus on endothelial dysfunction. I would encourage greater focus as well on sort of longer term vascular damage, which could potentially include damage to the extracellular matrix that helps to support the health of the endothelium. My hypothesis is that's really what happened to my daughter, was a sort of a massive inflammatory event that led to vascular damage, and her recovery over the last 4 years has really been a healing process, gradual healing process from that damage. But again, we don't know for sure, one patient, but would love to see more research on this. So you have this increase in vascular permeability. Now this happens throughout the body. It has really important effects. But in the brain, what we're focused on is a leakage of protein-rich fluid from the blood vessels into the extracellular space. And this is going to generate excess fluid in the brain. I don't know if this is the same as what Dr. Houghton was talking about with increased swelling. I don't quite understand the term, but if it is, then we're aligned on that perspective. But you basically have an inflow of excess fluid. And so you have certain people who have spinal conditions that inhibit the outflow of cerebral spinal fluid. And this increases intracranial pressure, and that leads to brainstem compression, as was discussed on the last time. And that surgery, when successful, reduces intracranial pressure by facilitating the outflow of cerebral spinal fluid. And that is one of the mechanisms. I'm not saying it's the only one, right? I would imagine that this leakage of fluid also irritates the microglia. So we can get back to, Dr. Renz-Polster's hypotheses around neuroinflammation. I also think, and this ties into, Athena, your point about the glymphatic system. We do observe glymphatic system dysfunction. We observe lymphatic system dysfunction. This is going to exacerbate the problem, right? Because you have excess fluid coming in. You have the glymphatic system either being overwhelmed or damaged or not working properly. That just is going to increase the amount of fluid that you have in the brain. So anyway, that's one hypothesis. Would love to hear some discussion about that. I would also just, since we have an extra 30 seconds here, encourage more focus on the effects of vascular leakage throughout the whole body. So for example, this is one of the hypotheses for lipedema. And Dr. Bluestein, you talked about this in a recent podcast, right? Is that you have leakage from the microvascular space. This leads to the formation of subcutaneous adipose tissue, which becomes fibrotic, right? And so there's that. You've got, I think personally that post-exertional malaise may be multiple phenomena, not a single phenomena. And one of those phenomena is related to the leakage of fluid that happens when you have an increase in blood volume and blood velocity, as happens with any kind of physical or mental exertion. And that may explain why my daughter has had more, was much more susceptible to exercise when she started than now, because she's had some healing and she has less vascular leakage and therefore less of a response to exercise. So a lot more to talk about, but we have only a few minutes, so I will defer to the next speaker. Thank you.
Thank you. Next, we're going to have Dr. Bluestein. So I'll mention, this time we didn't have a talk from the clinical side like we did last time. That's partly because we're going to be having a clinical roundtable with Dr. Yelman. I think it's December 6th. And so we're going to have a extended conversation on the clinical side there. But Dr. Bluestein is another clinician, and we have some other ones in the audience. So just wanted to make it clear that even though we're focusing on the science with the talks, that we're open to clinical conversations here today too. So Dr. Bluestein has been practicing medicine for over 20 years. She's an integrative medicine physician who takes a unique approach to the evaluation and treatment of patients with EDS. All right. You're muted right now. Oh, you're muted, Dr. Bluestein?
I had to stop sharing in order to see the unmute button. Sorry about that. Not my first rodeo, but you would think so. Let's see here. And thank you, Jeff, for the kind mention of the podcast. Can you see the correct slide now, do you think?
Yes.
Yes.
Okay, great. And I was very pleasantly surprised. Well, I shouldn't say surprised, but to have people mention mast cell, because that's basically what I'm going to just talk briefly about mast cell and the potential role in tissue integrity, the extracellular matrix, and the initiation and maintenance of pain, which are common problems with people with MECFS. And in my clinical practice, I was just really shocked that pain, of course, is the number one concern that people have, but fatigue is definitely number two. So just a little background for those who need a little bit of a refresher. Mast cells are immune cells that reside in the tissues near the blood vessels and nerve endings and are present where we interface with the environment. So the GI, GU, skin, and mucous membranes, and respond to external threats. Most commonly, we think of pathogens and chemicals, but they also have mechanoreceptors receptors on their surface. And these mechanoreceptive mast cells express TRPV2, one of the 6 transient receptor potential valenoid channels. And these moderate mast cell activation in the face of physical stimulation that can include shear forces, thermal change, and friction. And friction we would see more of with joint instability, of course. And also with things like barometric pressure and also tissue hypoxia, which I know was mentioned in a previous talk. So also in the periphery and in what happens in the central nervous system, we get more, I don't know if you can see my arrow here now. So this is the periphery and this is what's happening in the central nervous system. Okay, so in the periphery, in the nociceptive or pain sensing primary afferent terminal, we get the mast cells coming out and degranulating and releasing pro-inflammatory mediators that include serotonin, tumor necrosis factor, nerve growth factor, histamine, and tryptase. And then these activate the nociceptor or the pain sensing neuron, which then has secondary release of its own neurochemicals, which then cause the activation of more mast cells, and the cycle kind of continues. So we get this feed forward loop where we get more and more pro-inflammatory mediators and sensitization of the nervous system in the periphery due to the recruitment of immune cells, including the mast cells and some other immune cells as well. And then centrally in the dorsal horn, which is the one down here, where we see the first order excitatory synapse between the neuronal cell on the left and then the non-neuronal cell on the right. And this connection between the mast cells and these astrocytes and microglia or non-neuronal cells. We also get sensitization there because mast cells degranulate in the first order excitatory synapse where this communication is happening. And these nociceptors also release inflammatory factors that activate the second order neurons and non-neuronal mast cells, including mast cells. And that induces neuronal activation via pro-inflammatory cytokines and chemokines. So we see TNF alpha again and serotonin as well. And then that activates the second order neuron also. And again, we get another vicious cycle that can contribute to sensitization in the nervous system. Also, the release of proteases further degrade connective tissue and contribute to a loss of integrity in the extracellular matrix, which contributes to more friction and more activation of mast cells, and therefore the feed forward loop continues.
Thank
That's it.
you. Very good. So let's see. Before we get started on the discussion and Q&A here, I just wanted to mention that we will actually be having a third part of this roundtable series in addition to the clinical one. So clinical roundtable on all the mechanical issues with Dr. Braden Yellman in December. And then we're going to have another science or more research focused meeting on tentatively January 10th. And in that meeting, we're going to have a summary of all the molecular findings that have been found in MECFS and work on those connections, which is the original goal of the roundtable. So, but I'll mention, you probably noticed we have a very diverse, wide audience here, and patients, caregivers, clinicians, researchers, and that's one of our goals with Renegade Research, is to bring people together and make connections, look at this from multiple different angles. So yeah, like I mentioned before, as part of that, where if you come up for the discussion, feel free to bring up a completely different topic than what's been discussed here today. So yeah, we'll go into the discussion now. If you want to come up and say anything, you can raise your virtual hand. And we'll start off with the panelists up here already. If you asked a question in the Q&A, depends on which ones were upvoted, I might have gone in there and asked you if you want to come up on video or audio. So take a look if I went and sent you a question in there. So we'll start with Freshta.
Oh, wow. I thought they're going to be the last one because there's so many questions here. Well, thank you so much for inviting me. Amazing talk. Each 5-minute talks could have been 50 minutes or maybe 5 hours, I believe. I just want to ask 3 questions, and actually not questions, emphasize and highlight. One, regarding what Jeff mentioned, the lymphatic drainage system, specifically knowing that these people with connective tissue disorder, they don't have a proper connective tissue that really support their lymphatic system. And we know we don't have heart while our vascular system, we have heart that regulates the blood flow. For lymphatic system, there is no heart. So everything is based on gravity. And then often people with connective tissue, they might be more predisposed to their lymphatic system, might not be drained properly. And this is where all the immune system and all the activities we are worried for inflammation happens. So they are going to be channeling through this vessels. And if there is a block somewhere, then definitely they're going to be more inflammation. And specifically, if gravity controls, then they're going to be more issue around the neck and the head in this patient. So bringing in people who are really expert in how to help a better flow for the lymphatic system might really benefit these patients, a low-hanging fruit. And it seems that Jeff already has seen the value of doing it for his daughter himself through trial and error. The other one is, another things that I notice is patient's frustration, how to drag physician attention to all the things has been beautifully described here, from CCI to other issue in the brain that might exist or maybe stem from issue within the flow of the fluid in our vessel and in our other system. I noticed some patients have bulge vein in their feet, in their hand, and in their neck. I am so shocked to see no physician pay attention to that, and I would love to take the brain of you here to see that if having bulge vein at early age, is it really pathological? Is there a chance that this could be part of the giving us some clue that these people might have some issue? Generally speaking, it's being observed in older people, but many young people have it, and physicians tend to rule it out that there is nothing pathological about that, specifically if we are talking about a bulge vein in the neck area. And I think regarding the mast cells, I cannot agree more with how this could be important for a wide array of symptoms this patient have. And we published a paper studying one MECFS HSD patient longitudinally, and the time there is a great correlation between symptom reduction, specifically neuroinflammation, and Th2 reduction and mast cell activation pathway. And we have seen that reduction in CCL2 and a few other Th2 related cytokines positively correlate with symptom improvement. I just want to say thank you so much.
Yeah, thank you. If I could comment quickly, thank you, Dr. Freshta, Dr. Jahabani. First of all, on the mast cell thing, I think it's important to remember that the mast cell mediators, it was great to see Dr. Bluestein's sort of picture, but another thing they do is increase vascular permeability. Several of them are very noted. You know, we've talked about histamine, obviously, bradykinin, but several others also have very significant effects, right, in increasing the fluid leakage. I do think we need a lot more attention on the glymphatic system, but also the lymphatic system throughout the body. One question I have is whether we know there are amyloid fibrin clots in the blood. Is it possible that when we have this leakage that some of that sort of amyloid fibrin leaks out into the extracellular space and actually clogs up the lymphatic system as well? We know that fibrin on its own can clog up the lymphatic system, but that tends to degrade over time. But if it's amyloid and it has problems degrading, maybe we have some of that same problem there. So that would be really useful to look at. The other thing is, you know, we talked about connective tissue disorders. I mean, I've noticed with my daughter that when she has a bone go out of place, there's all this, you know, muscle, you know, spasms. We all know about that, right? And it spreads through the fascia, but it actually seems to impair the lymphatic system as well, which is very close to the edge there. And so I wonder to what extent there's, you know, sort of spasms in the fascia and the muscles that are impairing lymphatic drainage as well. And to the extent that the CSF and the lymphatic system is all one system, that's going to affect the brain as well.
Well, and there was the question about bulge veins or comment about bulge veins and circulation. And if we think about it at just sort of at a micro unit level, or sort of what's a core unit, if you think of one side of that unit being blood going in and then blood going out, arteries and veins, even if little capillaries, and then you have tissue in between, and then you have lymphatics to take away the waste material or things that need to be recycled, and you have tissue resident macrophages or some of the white cells sitting in the tissue, and you have mast cells sitting in there, and then you can get things coming from the vessels through those leaky walls. I think that's the circuit that's all getting talked about here. And so if you're plugging it up with whatever the material is, if it's microclots or something else, then if you don't have good flow at a micro level and at a brain level, that it's getting that flow going is critical to keeping oxygen going. And if you don't have oxygen going, you're going to increase inflammation, just the way the body tilts in a teeter-totter.
Thanks, John. And I just remembered I was going to ask people who come up and ask questions to do a very quick intro of themselves, just so we can kind of meet each other in the audience. There might be some people that want to collaborate afterwards. So I'll just mention Freshta is a researcher at Stanford working on MECFS.
And I just want to, I study MECFS overlap with HEDS, chronic Lyme, as well as PANS or pediatric onset, acute onset neuropsychiatric disorder. And the reason I do that is because my journey with MECFS started in 2013 when a family came to our study and they had all these conditions together. And little by little, it seemed to me that often patients have co-diagnosis. So we don't know this is comorbidity or really one disease start from one point and get to the next point, you become more predisposed to chronic Lyme, or the other way around. You get chronic Lyme and then you get HEDS or HSD to be exacerbated, and also comes with neuropsychiatric conditions in some population. So thank you. Yeah.
Thanks, Freshta. Another clarification. Isabel told me that Dr. Yelman isn't only talking about the mechanical issues that'll be part of it, but it's also going to be a broader talk about his clinical experience and how he treats patients. All right. Panelists, feel free to raise your hand if you want to come up. Freshta?
I would really love to know that here, because one of the purpose of this program is to help patients. What are our ways to, at the end of this roundtable meeting, because it has great power, having everybody here, how we can summarize and add more document to patients when they listen to this podcast. They can actually take this references and citations to their physician and have them to really value some of the symptoms they're having rather than being constantly underestimated or undervalued. From all this discussion here, I think one problem is that there have been many of this discussion in the field, but patients really struggle to go to a cardiologist and tell them that we have issue with our vascular system that is affecting the blood flow to our brain. Doesn't mean that when we say we have it, but there should be a way between patients who experience a wide range of symptoms and they know so much about their body that when they say something, it should be valued by the medical team to start from somewhere. But then the first test is they look at their cardiac structure and the cardiac structure is healthy. And they say, that's it. They forget about the element of cardiovascular, neurodevelopment, immune neurocardiovascular, and how they interact with one another. Maybe adding pictures, image, citation that they can take it to their physician after listening to this great podcast can really increase the impact of this great work.
Yeah, that's a great idea. I don't want to volunteer our medical director, John, but I was thinking maybe we could coordinate some sort of handouts or summaries after this with some of the clinicians. Athena?
I was just going to comment, indicating that when you had your earlier talk test, way back when with Dr. Ruhoy and Dr. Kaufman, I found that was very useful because I actually started to develop a document myself independently that I could prepare for my doctor, where he talked about the septad and his correction of the septad, where the 5 overlapping comorbidities of 7, for example, MCAS, CCI, and so on, so on. So that was very useful. I think if we could type that up and link it to the consensus criteria, if that does occur, that would be a great handout that I'd be happy to be involved in building with Dr. Houghton.
That would be awesome. And I just realized one of the things we're trying to do with Renegade Research is partnering with other organizations and make, if other organizations have already done this sort of work, sharing it. So I'd be curious from the audience or, from the attendees, does anyone know what sort of work has been done on this already? I know Bateman Horn has a lot of great resources related to diagnosis and treatment.
Well, the figure from Dr. Renz-Polster's paper with the feedback loops it's an incredibly powerful and complete picture that's very digestible, even if somebody isn't totally versed in all the biochemistry and everything else. And that may be part of the be useful if somebody's taking it into their primary care. And it's basically saying, these things, one thing jumps on another and you get these positive feedback loops, and you're going to have good days and bad days. And I think it's really important because you see somebody on a good day and you go, oh, there's nothing wrong, but there's a lot of underlying stuff happening.
So you just transitioned me into a question because someone has a question about good days and bad days. So it was in the Q&A. The question was, Herbert mentioned often good and bad days concerning PEM. I would like to know, what does Herbert think about severe and very severe, where you have exclusively bad days with 24/7 symptoms from hell without overexertion? A lot of patients are never without symptoms, so to speak, in a constant crash. And thanks for doing the talks.
Well, even the moderately affected are never without symptoms. No, not a single day. But for me, I think that it's a matter of threshold. Basically, I consider the severely affected and the very severely affected being in a constant PEM. They never can have the resource to work out of. They're constantly basically in a state of post-exertional malaise, because the threshold for them to go into overexertion is so low that they don't have the normal. They hardly ever reach their baseline, or the PEM is their baseline. Let's put it that way. Yeah. That's my take.
Freshta?
Okay, I'm so sorry to have too many comments, but this is something I am so passionate about, and I think there is a great area of need and research into this. When we talk about longitudinal study, and we are trying to actually come up with a way to measure how patient responds to a clinical trial or drug or stuff like that, we really need to understand what is the maximum level of suffering and what's the minimum level of suffering. To begin with, for MECFS diagnosis, patients can have a diagnosis if the symptomology they are talking about has led to the reduction, 50% reduction in their activity after the onset of the disease. Imagine you should be losing 50% of your capability of what you had ever before to say you have a mild form of MECFS. I highly disagree with this, because if we lose 50% of our functionality in personal life, social life, professional life, and everything else, that does not mean to be mild. We have to redefine the criteria for the mild to begin with. And then from there, we have to let the patient know what's the maximum level that you can lose your functionality, to know where they stand. And from there, we get the help from patient to give us objective way to measure when we provide them a medication or a group of medications together. And this really going to help us to say that now this is working or this is not working. I think this area really needs a lot of optimization. Again, in the paper we were publishing, it occurred to me that I see the differences, but the patient is still extremely bedbound and extremely severe. When you're still bedbound, to the outside world, nothing is helping you. But if you're bedbound and you cannot use your phone, and you're bedbound and you can use your phone and not be purely isolated from the social life by having at least access to your phone and internet, there is still an area of improvement. So when you ask patient, they might tell you, I'm really extremely severe. But if they don't have a way, objective way to see where they stand, that kind of really eludes to whatever data we are generating to find out what's going to help this subset the best and the other way around. And I think talking about the clinical trial, I think that was John was mentioning about this cocktail of how all the medication and remedies that they could provide the patients to get better. And this is another issue, that clinical trials are mostly meant to just work on one treatment, not multiple. Rarely we find a combination of 2 or 3 medications to be offered to the patient cohort versus the placebo to see what's happening. But the disease that is multisystemic needs multiple treatment at the same time to address the root cause, from the lymphatic massage plus this, plus antiviral maybe, plus anti-mast cell, plus this and that, to see what's happening. I would love to ask John how we can convince the clinical trial team that this group of patients needs multiple medication, not one.
I think it's in trial designs, and I think the kind of the idea of distributed trials and using appropriate observational information that can have controls by looking at real-time medical record information, for example, can allow you to find groups that have done combinations of things or individual things. But it's certainly the cancer world is a place where there's a multi-decade example with extraordinary results compared to not doing it. We're doing multiple things that are complementary and follow the biochemistry, make a huge difference.
Actually, the cancer ward, I have to remind that I work with cancer patients as well. And I do know, I might be mistaken, that even the board of cancer clinical trial are really limited to the number of because they want to really see the cause and effect, am I right? So the question is that there is a crazy growth of a cancer, so they mix max 2 or maybe if they could 3 medication together, not a wide range of. So I feel that this has to be discussed. Maybe we can think about it and just start from a base in placebo and add 1 more to the top of that, to the group that are getting a medication to see what's the impact. But one treatment for a multi-system condition might not necessarily help. There could be magically some subset benefit from that if we know what's the root cause.
Yeah,
But
if you have a large enough group, you could do a factorial design, right? Where half the people are getting one, half the, you have some overlap, right? So you have some people getting just one, some people getting both. And there are ways to design that so that you learn enough about each individual one and also about them in combination to generate reliable data. So I mean, that's something certainly that we use in the social sciences, and I would imagine it's used in medical trials as well. The other thing I would note, this issue about longitudinal trials, and I appreciate Dr. Renz-Polster's comment about the need for that. But a lot of patients are too sick to go into a clinic and be measured and get all their blood drawn and all this time. And so we really need to think about how do we incorporate patient measurements or measurements that are undertaken by caretakers. I think if that's done prospectively and carefully, there's no reason that they cannot be enlisted as contributors to the data collection. So we need to think outside the box. Otherwise, we end up with bias studies that omit the most severely disabled people from the study.
Yeah, very important point, and that's something that we're considering and really taking into account with our study designs with Renegade Research. We do a lot of sending at-home test kits to people, and we want to make sure that people aren't left behind. And related to Freshta's comments about testing more than one treatment at a time, that's also something that we've been doing with our studies. And I think one problem with that is funding. The funding agencies do just want to test one thing at a time. So I think we also need to be thinking about alternative funding.
oh my gosh, we can talk about that funding issue forever. This year, DOD's MECFS study received $300,000 for the clinical investigator award, only $300,000. And I don't know who is that lucky person, but it's.
Yeah. All right. Patricia?
Yeah, I just to chime in with what's being discussed now, when and the with what Dr. Houghton has presented, my own experience very closely paralleled everything that he is proposing, and working with Remission Biome and doing things on my own. And so I experienced a significant improvement. So there is a lot of value because I was bedbound at one point, and just working through and doing all of that, and things that you presented, Dr. Houghton, could be done, like Jeffrey Lubell was saying, for people who can't get to be tested and that type of thing. I actually implemented unknowingly or parallel to what you presented, and did experience positive improvements. So, and that's just echoing what we're just all talking about now. So I just wanted to.
Well, and just a comment about that. Some of those ideas, at least for me, came from a survey that was done. It was an online survey that somebody did about 3 or 4 years ago, Martha, who ended up finding things that are breakdown microclots, like nattokinase and others are important. And it was around the time when the idea of microclots in long COVID was an issue. And that absolutely drove some N of 1 personal experiments and kind of launches the stuff forward. I think there's so much wisdom in the lived experience, and how to harness that is critical.
Yes, agreed, absolutely. And I'm pretty confident I participated in that survey that she had put out. And again, use that as a guiding factor, kind of piecemealing everything, putting different pieces of research together to see what could be used in conjunction with how my doctor was treating me with IV therapy. And so, yeah, definitely that broad-based community knowledge is incredible and very, very resourceful and essential, really.
Great, thanks. Roshan, and can you just like 1 or 2 sentence intro of who you are, what you do?
Yeah, sure. I'm a researcher in the biotech industry. We develop immunomodulatory drugs. I'm also have been a caregiver for my wife who had been sick with MECFS for quite some time. And we do some research on MECFS with Lisa Celine's group on the immune system. I just had a comment on trial design, maybe coming more from an industry drug development perspective where, of course, there are traditional structures for running clinical trials. I completely agree they're too rigid and make it quite challenging to test combinations at times. And we work in cancer and autoimmune diseases, and I think certainly, well, in both contexts, there's a lot of instances where you might really only expect a combination effect. And not necessarily a monotherapy, but there's a lot of kind of inertia to test things as monotherapies first, for a lot of reasons, and they're good reasons at times, including safety is a huge one, right? And if it's any new kind of treatment, you must always have to test it in monotherapy to demonstrate safety. So it's much more feasible when you have either approved reagents or things where there's a very clear safety profile. Even then, the combinations are usually, adding one thing at a time. And then the other is seeing an effect and seeing an effect size. And, ultimately for industry, that's how you get your approvals. You demonstrate the added benefit of adding your single therapy. So I think we have to recognize that, and if we're thinking about large scale trials and also getting more industry involvement, there has to be some sort of acknowledgement of the existing structures. There are a lot more innovative trials now, including in cancer, with these kind of basket trials where you enroll patients, make a lot of measurements, and then sometimes parse them out into different subgroups being treated with different combinations of treatments based on their profile. But I think it's more tractable when you have, clear objective enrollment criteria and then things you can measure that are mechanistically tied to the interventions you are doing. Because even if it's multiple treatments where you have an idea of the pathway it's modulating, if you can then, connect that to something you're measuring, I think it becomes more interpretable. That's kind of hand waving, but I just want to add that perspective.
Yeah, no, that makes a lot of sense. It's helpful to have the different perspectives. So yeah, Fresha, is it related to that?
Yes,
Or I was going to
exactly.
read a question. Okay, cool.
Yeah, exactly. I just want to say thank you so much for bringing this on. As I was thinking, I think because in the real world, majority of the physicians treat these patients with a wide range of treatments. It's not just one single 2 drugs combination or 1 or 2 supplement. They come up with a tray of medications. So how we can actually translate it to the real life of improving and narrowing this long list that patients have to take on daily basis itself can really help patients reduce some of the liver toxicity that might come with it and make the life easier. And I'm sure eventually this challenge has to be addressed because it's a multi-system and physician using multiple drugs. So we have to talk more often about that.
Thank you. So I'll read a question from the Q&A now, because it kind of brings us back to the brainstem and thinking about how effects in the brainstem might affect molecular or their effects in the body. In Ehlers-Danlos syndrome, Dr. Claude Hamonet found dopamine helped 66 to 75% of the patients who had dopamine responsive dystonia. Is there anything about what was seen in the brainstem in MECFS research presented that might fit with DRD, and if so, might be the mechanism? This might be a question for us to bring back to part 3 in January, but just curious if anybody has any thoughts on that. Elizabeth?
Yeah, thanks. Can you say that question one more time? I might have a thought and I just read a paper.
Sure. In Ehlers-Danlos syndrome, Dr. Claude Hamonet found dopamine helped 66 to 75% of the patients who had dopamine responsive dystonia. Is there anything about what was seen with the brainstem in MECFS research that might fit with DRD?
I don't know about the brainstem, but I was reading a paper before I got on this call, the Avinath deep phenotyping of MECFS. Avinath is director at NINDS, and my name is Elizabeth Weaver, and I work at Georgia State University. I'm the associate director of brains and behavior. I'm also on the board of ME Action, and welcome any thoughts there around that group. But yeah, I was reading that paper, and there was lower amounts of DOPA and other metabolites in the cerebral spinal fluid of people with MECFS, and I thought that was really interesting. And they didn't talk about the brainstem in that study, which I also thought was interesting because they did talk about the temporal lobe and its relationship to the motor cortex, but I have a feeling that they left out the brainstem, and I think that's a really important part of this puzzle. And so I'm really glad to hear you guys talking about it today. And I think the temporal lobe, if y'all are familiar with that, I think the temporal lobe motor cortex thing they saw in Avinath's big MECFS study, to me, and I don't know if other people feel this way, seemed almost like a decision-making study. Like it was the decision of not to do something because you know you're going to be sick. And I don't find that super, that decision making very helpful to understanding MECFS. I think it's a smart decision, probably not to hurt yourself or do effort that's going to hurt yourself later. But those are my thoughts. That I think dopamine does have some sort of role here. I'm not sure what it is, but that study is worth a look.
Yeah, really interesting. Do you mind putting that in the chat, or yeah, I guess you can put it in the chat now that you're up here as a panelist. Do you have that paper handy?
Yeah, sure. I can put that in chat in just a second.
Okay, cool. Kiran?
Yeah, just brainstem do have a dopamine nuclei, especially in the midbrain. So yes, yeah, for example, substantia nigra, which is located in the near to the midbrain. So it's in dopamine, it exhibits or inhibits the dopamine that channel. So there could be some involvement in the brainstem.
Okay, cool.
But I don't have any research whether they have seen that one. But definitely, as we are talking more on the brainstem dysfunction or impairment, that could be the one reason. But yeah, have to do more research focusing on the dopamine only.
Yeah, that'd be awesome.
I hate to be a hammer with everything looks like a nail, but I do think, if you look at vascular permeability as a mediator here, dopamine certainly inhibits vascular permeability. So that may be one of the mechanisms through which it has an impact.
Thank you. Bresta?
Yeah, backing this, there are multiple potential ways, including the antibodies against dopaminergic receptors have been found in a subset of the patient's blood. We do know that mast cells release dopamine, and when they are being too overactivated, there might be a deficiency of depleting of the dopamine within the mast cells in a normal physiological system that they could release. I was going to say that there is a possibility with the substantia nigra in the brain of these patients might be affected, where we are having a lot of dopamine expression. And then dopamine itself can actually, when it binds to the dopamine D3 receptor on mast cells, can suppress mast cell degranulation. So I'm sure there are many other things going on, but one can think of multiple possibility. But I would suggest low dose dopamine modulator rather than drug or like Abilify, that at low dose they act as partial agonists for dopamine rather than those that at high dose act as antagonists. I think this group of patients might benefit the most from the least amount of the drug and mostly partial agonists than a full antagonist or pure agonist.
Cool. Kiran?
In case of substantia nigra, so it has got a high iron density actually. So if that's the case, maybe, there can be some relationship between the dopaminergic energy and the iron also, because it has high amount of iron over there.
Interesting.
So maybe we need to quantify the amount of iron and see the relationship with the dopamine.
Yeah. Might as well ask another question for you now that was in the Q&A.
Yep.
It says, how often did you observe obvious anatomical abnormalities like Chiari malformation in your MECFS cohort? Did you look at stuff like that? Any of these structural issues, or were you really focused on the specific?
Oh, we are very specific on the, actually we do study the whole brain, but every time we study the whole brain, something pops out in the brainstem. So we are very excited. Okay, guys, is there something related in the brainstem of MECFS? And we started more focusing on the brainstem. So all our MRI data acquisition is mainly based on the brainstem. So we try to optimize our data on the brainstem. That's why, like, our results might be a bit different from the other, because we are mainly focused on the brainstem only. So that's why, like, if we acquire the ultra high field 7 Tesla MRI, our focus was on the brainstem. So that's why we can see our results are pretty much reproducible from the different imaging modalities. So we are also doing now the longitudinal study. So I think that will somehow confirm whether the brainstem, like, is, we can track the changes in the brainstem. So I think that will give us the better idea. Is this a very consistent or the one-time effect?
Yeah, it would be super interesting to do that study though, Marissa. Yeah, like having somebody who's really a specialist in the mechanical issues go and split it into subgroups of who has each mechanical condition, and then look at if you see differences in each of those groups. Yeah, let's see. Anybody else want to raise their hand while I'm kind of looking through the Q&A here?
I think there was one question for me in the chat about is the functional connectivity or something like that. Did I miss that one? From the John, maybe?
Oh, I had asked a question about when you talked about more limited connection or stronger connection, whether that was speed of processing or number of connections. So basically, could you have connections that are demyelinated that might be processing more slowly, or is it a difference in connections? And I don't even know whether that's something you could tease out.
Oh, it's very hard to say it's a demyelination, because most of those regions are in the gray matter regions. So where do we expect the less amount of myelin? But what the functional connectivity tell us that there is some kind of degree of correlations between the two different brain regions. So maybe are they properly synchronized or not? So for example, let's say if the bones and the midbrain communicates with each other, then we can say, oh, the other is synchronized, or one has to work a bit harder to compensate for the another regions. Or for example, if bones is working harder, then because there might be some compensatory mechanisms because the midbrain can't perform.
And just a quick follow-up question. So as you're saying that, it makes sense to me, but I'm not sure it's right, that if you had faster transmission on the connections, you would have more synchronization. So you'd have what would be considered a stronger connection in what you're describing, if the connection is the correlation between the two parts. And if you had inflammation that was slowing down the speed with which nerve signals were propagating, whether that would then lessen correlation, because you could end up with different levels of delays on different neurons.
Yes, but I think we have to understand now the fast transmission is not always the better. The fast is not better. Sometimes if you look at in long COVID findings, we found the stronger connectivity when compared to the healthy controls. So the faster or the stronger connectivity is not also good.
Right.
So we have to be very careful here. So it's just a degree of correlations between the brain activity performing between the two different regions.
Thank you for explaining that.
Great. I've got a question or a comment in here from Deb. There is a recent article by Henderson, Rowe, et cetera, which follows patients who had craniocervical fusions and tracked symptoms, including ME-typical ones. So more data than the Rowe case study. And we just put that, or we're going to put that in the chat. Troy?
Yes, I have a question for Kiran Thapaliya. I wanted to clarify the point. You said that the pond size has an inverse correlation with the pain.
Yeah.
So the larger pond size, there's less pain.
I think it's again it's a degree of relationship or association between the pain and the bone's volume. It's not the cause and effect. So we have to be very careful with the correlation. It doesn't mean that if we have the high volume, you have the less pain. It's not like that, because there's no the cause and effect. It's like this some relationship. So what we found here is that if you have your severity pain level is higher, so that means there could be some kind of association with that, with the degree or the decrease in the bone's volume, but it's not the cause of pain that gives you the that create or that affects the bone's volume. It's just an association, and we have to be very careful here again.
Yes, I understand that is a correlation. I just wanted to make sure that I was understanding the correlation correct.
Yes. So that is that in that the correlation is less volume correlates with the higher pain level.
Okay. And so could that be because over time, as people increase neuroplasticity, that
could be, yes,
would
possibly that
increase
could
the
be, yes.
size of the pons, and that helps deal with the pain?
Yes, that could be, yes.
Okay. All right. Thanks.
All right. So I've been scanning the questions in the Q&A, but I think it would actually be easier if people just started raising their hands from the attendees. If you have anything that you want to come up and ask, we'll move you up to the audio video. Troy, did you have something else?
Yeah, I just another thing is the idea of the brainstem. Is it formally considered parts of the brain in most research? Because I was looking at the EU 3D project that they're putting together, where they're trying to have this open source platform of all the data and images for people to share. But when you look at the image, the brainstem's not there, which seems kind of strange that this open source brain database wouldn't include the brainstem. So I'm not sure if it's formally considered part of the brain, or is it kind of like a, I don't know, side thing?
I think it's part of the brain, but why people are not including that region could be like different reasons. Where one of those could be like some people, the project depends on the hypothesis. Maybe they don't have the hypothesis depending on the brainstem, and it depends on what kind of MRI scanner they use, because in some of the MRI scanner they might get the less signal to noise ratio, which won't give you the clear pictures of MRI, brainstem, sorry. So that's why we have to be very specific to get the better quality image of brainstem.
So you need a 7 Tesla.
No. We can do it on a 3 Tesla MRI also, but it depends on the project hypothesis that we do actually. So do we really need the brainstem or not?
Okay, thanks.
Yarrow?
So I was just going to add, since this involves the cerebellum, and we have just the comment about this low-lying tonsils in Chiari, I believe it was Dr. Dowdry with ASAP identified that 4% of the population have low-lying tonsils, but may be asymptomatic. And of course, it's not relevant unless patients present with symptoms or perhaps have a cyst or syrinx, for example, in the spinal cord, have an accident. Is there a relevance to your study of these patients who are predisposed to this potential issue, should they become symptomatic later, whether viral etiology or trauma or something else?
Yarrow?
Hi, thanks. I'm wondering if anyone could talk a little bit about brainstem seizures, especially differentiating them from epileptic seizures.
Do we have anyone fresh up?
That's a wonderful question. And I would say that we should distinguish this is metabolic driven seizure, or this is really hypoxia, lack of oxygen delivery seizure, or it could be increase in the cranial hypertension. So while I think this is an amazing question, but there should be some extra information and testing be done. I do have 2 patients that they have CSF leak, that they have seizure, and their seizure is being triggered when they are being overstimulated, stimulated with stimuli, and seems that the CSF leak, addressing the CSF leak has been helping to a great degree, still not fully removing this stimuli causing seizure. So I would say this is an amazing, again, area, maybe another roundtable to be dedicated by some patients have seizure, all possible causes, and then how physician can help this patient to really get to the bottom of their particular seizure, which seems not to be typical seizure.
I guess the second part of my question would be, thank you for your answer about seizures due to a mechanical basis of the brainstem.
So I think in general, seizures are extra electrical activity in the brain that could be rhythmic or what have you, and they are either focal, so they start in one place, and they can spread, or they're generalized, and then they have an amount of time they're associated with. And so if it were to start focally in the brainstem, you might have more symptoms related to things that are associated with the brainstem, versus if it started in the top of the head and a little in the front, maybe you would have something related to feelings in your hand or something as a partial piece of it.
Thanks for that answer. I actually have epileptic seizures and am very versed in epilepsy. My question was specifically about differentiating. I have had seizures related to CCI and traction and possible tethered cord. And I've seen so many other patients talk about their brainstem seizures, and I've had my neurologist say that was not an epileptic seizure.
Yeah, I've seen some people talking about that too.
Thank you.
And I'll also mention migraines with brainstem aura, which I have. So yeah, you get symptoms beforehand that like almost look like a stroke. and I've had one of the expert MECFS clinicians say that she sees it and seems to be related to things like CCI.
And you can get complex migraines that can look all the world like a stroke as they resolve. They can cause paralysis and stuff.
Anybody else? Oh, I just saw one of the question was, how do these MECFS findings relate to POTS? Are there differences in the number of men versus women in these studies for MECFS? Are the studies largely women? And I think I saw, John, you said you wanted to answer that live. Is that right? Or did you accidentally click that?
It was not knowing how to do the question stuff.
Oh, okay. Does anybody have any thoughts about that? All right. Anything else? Maybe we're slowing down here, coming to the end. Troy?
Yes, I had just one comment for Jeffrey Lubel. because he was mentioning the problems with permeability of the vascular system. And there's one of the research studies on nicotine with the mice showing the protection of nicotine during COVID infections. They hypothesized that the nicotine was helping protect the blood-brain barrier so that the different inflammatory molecules or the virus wasn't getting into the brain. So that's kind of one connection with what Jeffrey was talking about.
Okay.
I don't know if he's still here or not.
No, he had to leave, unfortunately. So I'll mention Beth Pollock shared in the chat that there's a EDS Canada Foundation conference tomorrow, and she'll be sharing some of their results on a hypermobility in these infection-associated chronic illnesses, and gave the link there in the chat. And then I see Janet came in, so I'll mention again that we are going to have a third part of these meetings, and we're going to really try to bring in some of the molecular basis researchers. We're going to have at least a summary of sort of all of the molecular results that have been found in MECFS, which is a ton. And so it brings up a lot of questions about how this all relates to, you know, Jeff said, you know, I forget the exact saying about the hammer and the nail, you know, everything looks like a nail, and we want to make sure that we also aren't, you know, so focused on the brainstem that we don't consider other hypotheses and, you know, put together the whole picture. So that'll be fun, and it's looking like we'll probably do that January 10th. So we'll send out information about that. And I'll remind people earlier, sorry to everybody who got my email like 2 days ago. I forgot to send out a reminder. All right, any other questions or comments?
I have one question, or one thing I just wanted to understand about connective tissue. When people say connective tissue disorder, for example, in hypermobility or EDS, which is connected to the brainstem issues of Chiari and CCI often, when they say that, what are they saying exactly is connective tissue disorder? Is it more a neurological thing, or does actually the biological components of the connective tissue?
Yeah, I'm not sure.
Does anybody have an answer or understand my question?
Looks like you're on mute, John.
Because I think Tess mentioned one time that when people are in a surgery and they're given anesthesia, then they become hypermobile. Basically, no? Didn't you mention that, Tess?
I don't think that was me. Maybe it was somebody else. And
Or somebody else. Okay.
yeah.
Somebody mentioned that. So I don't know if that's true or not. That would mean that it's maybe more of a neurological issue than the actual substance or the buildup of the connective tissue.
If you were to have anesthesia and had full relaxation of muscles, you certainly could would it make sense that you could have more hypermobility.
Mm-hmm. So
just
EDS,
from muscle tone, just changes in muscle tone.
So the solution for EDS could be more looking at the neurological side than maybe looking at the connective tissue makeup per se, like the biological side of connective tissue.
So some people, I think you're already well aware, some people have connective tissue that is more elastic and more loose. And so, and Ehlers-Danlos is part of that. And so that could be weighing in the side of more mobility of the bones. You could also presumably have inflammatory processes that can break down some of those soft tissues further, and that would make them less strong and less rigid. Likewise, you could have inflammation that would lay down fibrosis that could make them more rigid. There's something called bamboo spine, that ankylosing spondylosis or litis, that is along those lines.
Okay, thanks. Because one of the people that did an interview about the nicotine patch, he said that when he was at his worst point, his shoulder was falling out of his socket, and he doesn't have EDS. But when he was at his worst, his shoulder would just fall out of the socket. And then when he started using the nicotine patch, that just cured up, and he didn't have that problem anymore within a week. So just makes me think that it's maybe kind of a neurological
Yeah,
connection.
I would go down if you dive into questions or see it again, I would think of trying to tease out differences in muscle tone. So in that muscle across joints will hold them together more mechanically. Like your shoulder is a very shallow cupped joint, and there are a bunch of muscles around it, and there are ligaments too, tying bone to bone, but there are muscles around it that kind of keep the whole thing in together. And
Oh, okay, thanks.
Oh, you're muted, Athena.
If we think about connective tissue diseases due to genetic defects, you know that they can cause a problem in the structure and the strength of the connective tissue, and they're not all the same. There are a different variety. There was an excellent article, if I can find it, published, and there's one particular, you could say, category that is linked to a higher prevalence of craniocervical issues of the group. So for example, we can have Ehlers-Danlos, we can have osteogenesis imperfecta, we can have Marfan's, and so on. They're not all characterized the same. The second thing I want to say is Dr. Bolognese's discussion about the understanding of EDS, and that's that not all EDS patients go on to develop craniocervical issues such as CCI. But once they do, Dr. Bolognese actually calls it a evolutionary advantage, which I say is kind of, would not be a popular view among EDS patients that are suffering. But that having been said, once an EDS patient develops any kind of biomechanical issue, this really budges the needle and makes the problem so much more difficult for them. So that's something to kind of characterize, is that the newer thinking that not all EDS patients will go on to develop biomechanical issues, but the ones that do are going to have a lot of issues get improving.
Yeah, that's a great point. Good question for people to research. You know, why everybody doesn't.
There's another thing that happens, and it may or may not be related, but when women are pregnant and going into birth, there are processes that relax ligaments, and it's conceivable to me that there could be some overlap of with what we're talking about here, but I don't know of any.
Anyone else? All right, last chance. All right, so we'll end here. We'll send out a follow-up email that has the recording of this, and then all the resources that have been shared in the chat. And then if you want to get on our newsletter, so you are notified when we have other events coming up, like this part 3 in January, go to remissionbiome.org, and the newsletter signups on the bottom. Anything else anybody here wants to share from our team or anything? Okay, we'll end there then. So thanks for coming. We'll see everyone later.